Okay, on this week's Arabidopsis Research Roundup, I'm really pleased to be joined by Emily Larson, who is now working at the University of Glasgow with Mike Blatt. And we're talking about a paper just published in Plant Physiology, the title of which is Clathrin Heavy Chain subunits coordinate endo and exocytic traffic and affect stomatal movement. So it's great to have Emily talking about this paper. So I must admit I haven't thought about Clathrin since I was a third-year undergrad, I don't think. Great to see that there's a paper about it and how it's involved in plant development. So, hi, Emily. It'll be great if you can give us an overview about the paper, please.
Yeah, so initially the idea for this paper came out of a collaboration with Annie-Mary and Pohl, who is the Clathrin component, really, of this paper, since my background is more in the snare part of this paper. But I've always been really interested in how proteins need to get moved around, or how anything really in the cell needs to get moved around, and that in plants with their multiple Golgi and with their cells that can't really move around, they've got to really be able to move everything else around as best as they possibly can, and it's got to be coordinated. But we really don't have examples to support that model in the end. So a lot of the ways that we look at how the vesicle trafficking pathways are coordinated, or how they function, is by using inhibitors and pretty harsh chemicals that are very well accepted tools, but in my opinion are also not very specific. They're kind of like hammers as opposed to scalpels. And I do have the benefit of having a bit of a genetic background, and so I really have a affinity for using mutants. And it also just so happens that Annie had our endocytosis mutant, which she identified in a really beautiful screen, where she did a suppressor mutant screen, which I always think are kind of fascinating anyway. So it turns out that her mutant, HOS1, it is a mutation in the Clathrin Heavy Chain 1 gene, which she had identified, and which really contributed a lot to this paper, especially within the design. And what I liked about this is that it's a particular effect in one subunit in endocytosis machinery. So instead of using a chemical to inhibit endocytosis, this mutant, we can really just affect the actual mechanisms of a particular endocytic pathway. And we know that Clathrin is very involved at the plasma membrane. So with what I had been learning about CYP121 working in Mike's lab, and knowing that there was that's a pretty strong secretion mutant, I thought, well, maybe we can learn a little bit more about this CHC1 mutant, but maybe we can also use it as a tool for understanding how endocytosis and secretion are linked together, and how that supports growth or any type of kind of cell development or function. And with the tools that we have in the Blatt lab, we could also really utilize this for understanding stomatal movement, which is something that we're also interested in.
So in the paper you analyze mutants in both copies of subunits of the Clathrin heavy chain, and you show that there's a really specific phenotype, a stomatal phenotype. So can you talk it through the phenotype that you see in these mutants?
Sure. Yeah, so those well-versed in Clathrin will know that there's a lot kind of already known about the CHC1 and CHC2 mutants in mostly in roots. And this is because a lot of the endocytic tools that we have, or the labeling tools that we have for endocytosis, is really easy to do in roots. This particularly has to do with the FM464 lipophilic diet, which I use quite a bit in my paper as well. So we know anyway that the CHC1 and CHC2 mutants have reduced endocytosis, and that this is basically done by throwing some FM464 on your roots, and then looking to see how much is internalized after some period of time. So that's how the Clathrin mutants were initially characterized. What we've been able to do in this paper is we've been able to actually measure the rate of endocytosis. So we were able to show that the CHC1 and CHC2 mutants have it's not that they don't have endocytosis happening at all. Mm-hmm. It's actually that it's really, really slow.
Okay.
So that kind of adds a little bit to what we already know about these mutants. And when I'll say that this paper was not accepted on its first round.
Okay.
Just so little transparency and a little bit about just like the regular process of papers. Mm-hmm. But one of the comments that I got from my reviewer, which was totally fair, was, okay, so the Clathrin mutants have this root phenotype, but you're looking at stomata. How do you know that there's an endocytosis defect in the stomata?
Yeah.
I was like, yeah, that's something that I should probably address. And I think sometimes as cell biologists we say, oh, well, if we see a phenotype in this one thing, we can just apply it to anything. And I think that I sometimes am guilty of that too. So that was a really good and useful comment that I received. And so one of those things was saying, how can I actually measure endocytosis in stomata? And I basically applied the FM464 labeling protocol that we usually do in roots, but this time in leaves. And it was a little finicky and time-consuming, but we were able to basically apply the same type of measuring protocols, which was being able to take an internal, a ratio between the fluorescence that we can measure at the periphery, so at the cell membrane, right, and then also on the inside of the cell, and then over time be able to see how that ratio changes.
So for me, I think you kind of buried the lead a little bit there, actually. So I think one of the really interesting things about the paper is that you show in these Clathrin mutants that the plants have a really surprising effect on response to water stress. So can you just give us a little bit of detail about how these mutants respond to water stress and why that was maybe a little surprising for you?
Stomata are a really interesting specialized cell type, in my opinion. This is because over the course of the plant's life, they need to change their cell shape over and over and over again. Mm-hmm. And they need to do this in a relatively regulated manner, and they need to do it kind of fast, too. And I think it's easy enough. I always kind of thought about them as balloons, that they get bigger when they need to open and they get smaller when they need to close. Mm-hmm. And I never really thought about this meaning that there needs to be more membrane added as they get bigger so that the cell surface area can actually get bigger. Mm-hmm. This also means that the cell wall needs to be reorganized, so that they can actually expand and then reduce. They kind of need to pull everything back in again when they close. Mm-hmm. So if you think about it like that, then it's not just that they're blowing up like balloons, that you actually do need to have new membrane, potentially new proteins, and definitely potentially cell wall components reorganized and added and subtracted throughout the course of the day, at the very least. So if you think about it like that, you can think that vesicle trafficking's got to be really important. And we already know from Mike's work that when you have secretion mutants, so the CYP121 mutant, for example, that their stomata do not open and close properly in responses to environmental stress. So when we saw that this Clathrin mutant had a very similar phenotype, or that had a stomatal phenotype, actually, we thought, well, maybe this is kind of a nice companion to, what we see on the secretion side, which further kind of helps support this idea that you need to have coordinated secretion and endocytosis of the plasma membrane so that these cells can function. But what we didn't know was whether or not the cells were, that the stomata just couldn't open and close, or if something else was, kind of developmentally wrong with that. So we have kind of stacked some evidence together showing that the endocytosis is affected in the Clathrin mutants, that secretion is also affected in these Clathrin mutants, which is actually the data that I was kind of most excited about, and that this is kind of correlative to the stomata not being able to open very well, and they also really can't close very well. And so In the initial paper that Annie's group put out, that our paper kind of builds upon, she shows that the CHC1 mutant, her mutant HOS1, closes in response to lower doses of ABA, which is kind of what got us onto the water stress component. She used ABA as a way of mimicking water stress. For us, we actually grew the plants without any water. And when we also grew them under different light conditions and different humidity conditions, what we saw really was that the plants just didn't grow very well when they were under conditions that really required a lot of gas exchange control. So they couldn't grow as big as wild type. And then the drought tolerance or the water stress tolerance is kind of, it seems like a positive, but it's really just a phenotype of a mutation of cells that can't expand or contract properly with respect to the GARD cells. And we think that this is because Clathrin is affected in both secretion and endocytosis.
No, that's key. I think so. I mean, it's really nice through the paper that you start with this cell biology, but then it tracks nicely to this more broad phenotype, and that potentially has some quite significance in the way that the plants can respond to environmental factors. So that's great. So thanks very much, Emily. You've really given us a nice overview of your thinking behind the paper and where it's kind of come from. And I think hopefully people have enjoyed listening to this. So thanks very much for joining us.
Thank you.
And as I said, there's a lot of potential directions this can go in. So we look forward to hearing where it goes. Thanks.
Thanks a lot.